So here's just secondary structure looking at strands and sheets and helices and turns and loops. So every protein then will have a certain amount of these or maybe none, and that gives some insight into function. Okay, a protein has to be functional, or why does the cell make it? Okay, it's hard to do a question mark here. So function. Function here, secondary structure. This is just showing the different structure here. So that's secondary, and that's going to lead to the tertiary structure here. So mutations that break helices or strands or alter loops and turns would have a dramatic effect on a protein. And so we'll talk more about different types of mutations here. Here's the alpha helix. These are said to be coiled coils. You may have heard that term, and they have a certain structure. Notice the R groups on one side, and there are R groups kind of on the other face. So there are special situations, probably in molecular we'll talk about, to where certain proteins have certain amino acids. Leucines is an example. There are proteins that have leucines on one face, and those are said to be leucine zippers. They actually bind to another protein that has an exact leucine face as well, kind of getting back to the interaction here. So leucine zippers. There are other kinds of structures as well. Just let yourself flow into this. We will go back and come back into specific areas. So you won't, this is not the first and last time. And again, I'm thinking you've had a lot of this. I'm just adding and decorating a few extra treats that you may not have heard. And here's some helical proteins. Our hair fibers are helical, showing the coil regions, and then the different filaments coil together, and their various cells. And here is a coiled coil motif, and specific amino acids, specific proteins that are bound together. So bioinformatics can define one protein as having a coiled coil. That means that its partner would also probably have some type of same domain. So when trying to figure out pathways, we want to understand how proteins come together. Excuse me. And that's proteomics. How proteins come together and fold and twist and turn and make these advanced structures here. Here's some more. If you've had any physiology or A&P, you know about the muscles, the sarcomeres, the sliding filaments. Those are based on helical structures here, and they're trying to show you the more advanced structures here of the muscle fibers, and you've probably seen this before. This is just some examples of proteins that have alpha helix as their main one. They actually have codes that are in the protein database, and what they're showing is a code, and then they're giving you 5 examples from different organisms, whatnot, that have that basic structure. So when you're searching the database and you see a protein has this type of structure, then you can kind of focus further on what it might be. So this would probably mean you've got a homeodomain, and that's probably DNA binding. So it's probably related to binding to gene promoters or other kinds of transcription factors here. And we'll talk about that in molecular. Okay, the homeodomain. So there's the word domain there. Here's some enzymes. Okay, enzyme glycotransferases, and there's a alter structure. That's a pattern for a lot of these enzymes. Here's some proteins that bind iron, and those are known as ferritin proteins. Notice they have a lot of, there's even an iron binding protein in bacterial ferrin found in E. coli. And then here's serum albumin, and of course we have this in us, and this is its overall structure. So these proteins have all alpha helix, and there's the structure as well. This is the opposite structure. Okay, so this is the opposite structure, known as beta sheets. And these, like I said, it looks like pleated sheets. Pleated sheets. Here's the pleats. I'm going to beat me some dogs here. Go on. Go on downstairs. Okay. So, beta sheets, pleated structure. Notice how the structures are put together. This is a lot of different side chains, along interactions. So again, a different kind of structural motif here that would point you to certain different proteins that were all alpha helical or coiled type of proteins. Here are some examples of beta sheets. Here's these beta sheets can form barrel structures. Notice that's why it's called a barrel. It's a circular. And here is antibodies. Note that antibodies, the heavy and light chain, you may not have got into that, but those contain, and you see those little ribbons, those represent beta sheets. Okay, beta sheets. And so it shows the heavy and the light chain. So this is heavy chain 1, this is heavy chain 2, light chain 1, light chain 2 in blue, so the blue versus the red. And so again, where antigens bind and other proteins, here's an example of another protein that can bind to antibodies. Over here you see that little cutaway structure. So when we're doing bioinformatics and we're looking at the structures, we're looking for primary first, then secondary, tertiary, and then quaternary. So we're looking at evidence of what the protein is, and that gives us aspects of where it might be, who it might be binding with, and potential functions at that point, especially if it's something new. And if somebody has a mutant version and there's a disease, we try to find the correlation then between the mutation, the protein structure, and the disease or the symptoms of it. And here are proteins that have all beta sheet structures. And again, you can see the databases will give it kind of a different name for the generic pattern, and then there's specific proteins underneath there. So if you go to the protein database, PDR, I guess it is, part of that in your search for proteins and learning about predicting structure, you will find these various codes here, and that's just meaning they're trying to give a generalized domain for the pattern they see, right? So if a protein has a certain pattern, that gives us indication what it is, and probably even more important, what it's not. And so there's an example there. And so some proteins actually have alpha and beta structure, and here's those patterns here, and they're showing that. So this is the protein database, so it's the PDB identifier. So that's called the PBD identifier, and then they're talking the fold structure, the subfamily, family, protein, and then species here, and showing the different how they classify, in these cases, proteins from E. coli, jellyfish, humans, and these have alpha and beta structure. So you can get a prediction of what proteins are and how it might be related here. Turns and loops then, and here's an example of a a receptor that is binds to G proteins. When we talk about signaling, we will talk about our friend G proteins. Okay, so that's coming back. Notice here's the N terminus. Notice here's the C terminus. And these are hydrophobic 20 amino acid domains. And this is known as a serpentine receptor. See, because, right? Looks like a snake coiled in. This is the lipid bilayer. Okay, so this is the outside, this is the inside. So we might receive a signal here, a growth factor, and then it looks like other proteins are recruited once that signal's been received. And you're saying, well, how does this protein transmit the signal? Well, we'll talk about that in more detail, but let me just say it's a change in structure. When the ligand is bound to the receptor, that causes a shape change, and you get changes, especially in the cytoplasmic domain. And that then provides platforms. Think of a airfield that is now opened up, and various other proteins can come in, bind, interact, receive the message, and then move on. And so that whole process is known as a cascade, okay, where you get a cascading effect of proteins interacting here, and they're moving down to eventually the DNA, right? You're turning genes on and off at this point. So from outside the cell, binding a ligand, right? Ligand, and trying to spell that, ligand, okay. This thing is sucky to draw. I need a tablet, but I don't have one, but maybe I'll get one down the road. So ligand binds, that signal is transduced, you get a shape change, other proteins come, such as G proteins. They then have proteins that they interact with, and so forth. So it's a series, kind of like a waterfall of proteins, to eventually you go to the gene promoter. If you remember the lac operon, there was a promoter that turned genes on and off. So you get up or down regulation of genes based on a cell receiving a signal. And when we talk a little bit about gene chips, we will talk about how people can measure every gene on and off based on a signal. That's where we're at now. When I first started, we didn't even understand the cascade. We knew things were happening here at the exterior, and we knew DNA was being turned on or off, but we could not figure out, and we not put the proteins together, kind of like Tinker Toys. We hadn't put them together yet, or Legos. Now we know the Legos, okay, that lead us all the way down. Okay, so informatics spots what's going on here. Informatics can spot what proteins are in the cascade. Informatics can predict what proteins are interacting with the DNA, all because there was some growth factor or some signal, right? And we get, and that's called cell signaling here, signal, well, cell signals here, cell signaling. Cell signaling. Oh, I'm off the page. Yeah, cell signaling. So just getting you ready for that, just warming it up a little bit. Here's tertiary structure. Finally got to that. And usually it's at the tertiary structure that we see the term domain. And bioinformatics software will take a protein, predict a structure, and then annotate or predict the identification of domains. And again, that gives you big insight into what the protein is. And so that's how fast we're able to move now. When we find a new virus, for instance, we can sequence the viral genome and have predictions of all the proteins in it and proteins that might provide a vaccine within a day or two, where it was years previously in the 70s and 80s. So we've really, the good news is the zombie apocalypse is going to really have to be something surprising for us to be overwhelmed by it. But anyway, so I'm hoping that we don't have to deal with that. So here's in the quaternary, the tertiary structure, where you have multiple chains coming together. Here's a receptor, and notice here, instead of one protein, we have 4 proteins that makes a receptor. Here's our hemoglobin, the alpha and the beta chains with the iron binding. Here's showing the viral proteins, and notice the viruses have a protein that sticks out, and it's like a rope at the dock. It grabs our cells. Here's the HIV. It's a CD4 molecule. So if you ever wonder, why does the HIV virus infect mainly white blood cells that have CD4? Because their protein on the surface happens to bind to that. That's why there's no CD4 receptor complex in liver cells, for instance, or in the brain, which is really great. If there is anything, imagine having HIV and it would attack every cell. That would be just horrendous. So this then allows us to make medicines to block this interaction, or uncouple that, right? Protect people. So a vaccine would produce an antibody, right? Kind of the V structure here. Here's the antibody. And so imagine this coated the virus all the way around. And now these little sticky proteins couldn't bind. They would just flow through us. That's where we're going. We would love for these viruses to just move on down the road. Okay, so that's where we're hoping that's going to happen. So protein folding, how do proteins fold? It's a very energetic, cooperative, and it showed unfolded, and then there is 50% of the protein is folded and unfolded, then finally it's a folded protein. So, and it goes in reverse. A folded protein becomes unfolded. Things like heat, breaking the secondary, tertiary bonds and structure, can then release a protein for further study here. And so structures can go through multiple steps, and it's said to be chemical intermediates or protein intermediates. And I know in probably chemistry you've gone through that. And certainly in biochem, you will have to hit that when you study enzyme plus substrate reactions here. Enzyme substrate reactions. And this is the beginning of helix formation and collapse, showing the amount of energy. And you have to kind of have energetic driving force to get, notice the amount of energy required here. And you have to get percentage of residues of the protein in native conformation from 100%, so completely folded here. And you have to deal with a lot of energy and biophysics, and that's not what we're dealing with, but I just did want to kind of remind you here. Here's our friend the ribosome, has R ribosomal RNA and ribosomal proteins, okay, plus R proteins. And that makes up this complex. And it's so big, it has the weight of a Svedberg unit, a guy named Svedberg. Remember the 40-60 versus the 70-30 in bacteria? So because bacteria's ribosomes are different, we have a lot of antibiotics, and that's good. Because if bacteria have the same ribosomes as we do, then antibiotics would try to kill us as well as the bacteria, and that's probably not good. So there are protein machinery that helps or finds misfolded proteins. Here's an example. And notice there's ATP required and used, and a whole complex of proteins that then release energy, and you end up with the protein properly folded. So this has to occur, and in humans, this goes best at 37. So if you ever wonder why they take your temperature, this folding stops at about 108. So if you ever wonder why people at 106, 8, it's bad because proteins will stay here and not go here. So your cell dies because the proteins are not available for their activity. Okay, so we want to stay in this region so that proteins will fold correctly. And if we get too hot, too high of a fever, we'll die because the proteins will be misfolded. And here's an example of a misfolded protein in Alzheimer's. Here's a protein making little fibers, and those fibers then block neural activity, and so kind of almost a hole formed here, and you end up getting low mental activity because of this Alzheimer's. And these misfolded proteins keep going all the time, so it's a progressive disease, and we would love to figure out a way to take the proteins and unfold them and then refold them correctly. So that would be the genetic therapy here. We're hoping that, so we'll talk a little bit about protein folding, mutations. All this we will cover again in some form. This is just setting up understanding proteins here. So that's the end of this part. We will see you on the final segment here in this lecture. Take care.